How to make alanine. How can we make alanine? What's its precursor? Pyruvate. How do we make aspartate? What's one way to make aspartate? What's its precursor? It's keto acid. Oxaloacetate. And what about glutamate? It would be alpha ketoglutarate. So see, you already have 3 of the 20 again. So easy breezy. All right. Most bacteria and plants can synthesize all 20 naturally occurring amino acids that are used to make protein. However, mammals cannot. We can only do 11. The others are called essential amino acids that you have to pick up from your diet. Okay. Unless, this is just a little caveat, unless, we say otherwise, we're going to be talking about it with respect to bacteria for the amino acids. There's 1 or 2 that, for example, cysteine, one of you didn't write your paper book, about where we'll talk about both bacteria and mammals because it's very, very different. Okay. So all of the amino acids can be derived from intermediates of pathways that we've seen so far. So things like the citric acid cycle with alpha ketoglutarate and oxaloacetate, or glycolysis, like pyruvate, or some of them maybe just 1 or 2 steps removed. Then we have a few that are very, very, very complex. And that's why they are the essential amino acids. Okay, so typically they split them up based off of their precursor. So, alpha ketoglutarate, it makes sense how we can make glutamate from it. And of course, we know how to make glutamine with glutamine synthetase. But we'll also talk about Prolene and Arginine. We have pyruvate, which does alanine. Valine, leucine, isoleucine are ones that are essential that we cannot make, but we have to get from our diet. We have PEP. Okay, PEP and erythrose 4-phosphate from the pentose phosphate pathway can make tyrosine, phenylalanine, and tryptophan. Technically, if you notice, tyrosine's got the little, the little cross instead of the asterisk. So we can make tyrosine, but only if we have phenylalanine. Okay, so that's why some will say it's an essential amino acid. Some will say it's not because phenylalanine is an essential amino acid. But yeah, if we have phenylalanine, then we can make tyrosine. If you're healthy, you can make tyrosine. Okay. And then we have those that are based off of oxaloacetate, 3-phosphoglycerate of glycolysis, and then ribose 5-phosphate. That's histidine. One common intermediate here is 5-phosphoribosol 1-pyrophosphate, which is called PRPP. I know it's a mouthful, but if you look at it, see, this is ribose. See, it has a monophosphate and a diphosphate, which, remember, diphosphates like this in biochemistry also come pyrophosphates. So that's why once you break it down, it makes sense. 5-phosphoribosol. And so that's why we have phospholipid pyrophosphate. Okay, and it is PRPP is an intermediate in many pathways. Okay, and so if we look at PRPP in greater detail, we get it directly, we get it from the pentose phosphate pathway. And so there at the bottom, I have the enzyme, the way for this to get it. So we have ribose 5-phosphate. It requires ATP, and we get 5-phosphoribosol 1-pyrophosphate, PRPP, and AMP. So what class or classes of enzymes can this be? Oh, it could be a synthetase because it does require ATP. What else could it be? Pardon? It could be a kinase because it requires ATP. We're adding. What are we actually adding to ribose 5-phosphate? What group? A pyrophosphate. And so this one's called the pyrophosphate kinase. But it is a kinase. And it does make sense that I can see where people would think, oh, it's a synthetase. Okay. not completely out of left field. All right. On your own, you know, the question was, because normally this is the beginning of a different day, is how, you know, do we get nitrogen incorporated into that? And it's glutamate and glutamine. Remember, if you had to guess. Glutamate from chapter 22, part 1, one that we just finished. And so I'm not going into any greater detail with that. Okay, so first up, we have the alpha ketoglutarate derived ones. I expect you to already know how to get glutamate and glutamine. Glutamate, what enzyme goes from alpha ketoglutarate to glutamate? Amino Transferases, okay? Then I would expect you to already know how to go from glutamate to glutamine, because what enzyme was that one? Right, glutamine synthetase. So we already know that one. So now we're just going to learn about proline and arginine. This is the big picture. Prolene and Arginine. We're going to break it down. Okay, and so it's scary looking, but it's not nearly that bad as what you think. Okay, so if we start to put in boxes, let me see, which one do I go to first? Okay, all right. So let's start over here. So this is glutamate, right? It just looks, maybe it looks a little different because it's laying on its side instead of being up and down like we usually draw it. Here, let me bring this down a little bit. So bring this down so you see everything. Okay. So we have glutamate. You have ATP, and you're going to you're phosphorylating it. But instead of adding the phosphate to this end, we're adding it to the gamma end. Okay, so that's why it says gamma glutamyl phosphate. What class or classes of enzymes can do this? It's what? It's a kinase. So if you're going to name it, what would you name it? Pardon? It's a kinase. But what specific kinase is it? What happened? See? What is that blank one? It's glutamate kinase. The next step is where either NADH or NADPH is used to transform the gamma glutamyl phosphate into an aldehyde. So if you look at the name may look a little weird. It's glutamate, gamma, semi-aldehyde, but it's an aldehyde. So what class or classes of reactions can do this? What class or classes of what enzymes can do this? It's a dehydrogenase. See, you already know this. So what, if you're going to name it specifically, what dehydrogenase could this one be? What do both of those compounds have in common? It's glutamate, so it's glutamate dehydrogenase. Please note that the next step doesn't require any enzyme. That's why it says non-enzymatic. It's related to the chemistry that we've seen with sugars. Remember how sugars can be linear versus like the furoses and purinoses and things like that. So it doesn't require an enzyme. But it's a proline group. Okay, that's where that name comes from. This is a proline 5-carboxylate. Okay, which they abbreviate as P5C. So P5C takes an NADH or NADPH and transforms it into proline. So what class or classes of enzymes could this be? Pardon? There's only like 2 possibilities. Dehydrogenases and reductases. This one it's the old traditional name of reductase. If you had called it dehydrogenase, I'd give it to you. But the proline carboxylate reductase, that's its substrate. If you called it P5C reductase, that's fine with me. Just like any other time that I've done this, you know, I may have a box around something, either a substrate or a product or the enzyme. maybe the cofactor, something like that. But I would give you the rest of the missing pieces, and you just have to supply them one. Okay. Any questions so far on making Prolene? Only that last one would be a little odd because they still call it reductase instead of dehydrogenase. If we go back up to the top, up here, we have glutamate, and we're making N-acetylglutamate, which is really similar to, remember, the N here is a locant, like what we've seen with like glucosamine, like N-acetylglucosamine, things like that. So we're adding an acetyl group to it from coenzyme A, from acetyl coenzyme A, I should say, and we get the coenzyme A back. Okay, so what class or classes of enzymes can this be? There are at least two that make sense. There's the transferases, and then there would be not necessarily kinase, because kinases do phosphorylation, but what else is there? A synthase, okay? And so this one's a synthase, because we're making something. If you had called it the acetyl transferase, then I'd give you at least partial credit, because it is transferring the acetyl group. What are we making? Yep, so we put it together, and it's the acetylglutamate synthase. If you said N-acetylglutamate synthase, that's fine. That's even more correct. More correct. I should say that's more descriptive, more precise. Okay, so now we go from, the N-acetylglutamate. We take an ATP, we're adding a phosphate to N-acetylglutamate, but if you notice it's analogous to what we've seen here. That's why they like to put them side by side to make N-acetyl gamma glutamyl phosphate, which is a mouthful. What class or classes of enzymes could this be? What? It's a what? Kinase. If you're going to name it specifically, you would name it. Pardon? It's N-acetylglutamate kinase, right? Because it's named right after its substrate. Nothing earth-shattering. We see the comparison between Prolene and ultimately what will become arginine. Arginine is just a little bit more troublesome. Okay, now the next step, we have to use NADH or NADPH to kick off that phosphate to transform it to N-acetylglutamate gamma semialdehyde. So what class or classes of enzymes could this one be right here? It's a dehydrogenase, and if you were going to name it, you would name it after the substrate, exactly, which becomes really, but it's N-acetylglutamate dehydrogenase. If you had written out N-acetyl gamma glutamyl, that's fine with me. You know, I would give it to you, so people know what you're talking about. Okay. Now we take this N-acetylglutamate gamma semialdehyde. We're transforming it to N-acetyl-ornithine. This enzyme takes a glutamate and turns it into an alpha-ketoglutarate. So what's the only difference between our substrate here and our product? Right, in addition of this. So what class or classes of enzymes is this? It is. It's amino transferase. Okay, and technically it would be, you know, an acetylglutamate or an acetyl-ornithine amino transferase, but if you gave it a specific name, I'd give it to you. Okay. This is where we still have not made arginine yet, and so it's amazing when you think about it. We take N-acetyl-ornithine, and what are we doing? We're transforming it into good old ornithine. So This one has a special name. Like if you had called it a deacetylase, I would give you credit for it, at least partial credit. That is the type of reaction that's occurring. Okay, but it's got a common name. I should say common because it's not a common word. But you know how whenever we've had things like enolase and aldolase and all that kind of things, arginase and all those, this one is N-acetyl ornithinase. Hard for me to say that as well. And then the box we've seen before. It's just the urea cycle. Because once you have ornithine, we can make arginine without having any issues. That's part of the urea cycle. So that first step right here of the urea cycle, well, technically not the first step, but this first step shown here from ornithine to make citrulline using a carbamoyl phosphate. What enzyme was that one? You just took the exam over it. Yes. What was that again? Yes, ornithine carbamoyl transferase or transcarbamoylase. Give it to you either way. Then we have citrulline. We take the citrulline. It requires ATP. Remember, the urea cycle showed this as two separate steps. We're now condensing. That's why you notice it says ATP plus aspartate. You get AMP plus an inorganic pyrophosphate. There's all one enzyme that did that to make argininosuccinate. So what enzyme was that one? Arginino what? It's argininosuccinate synthase? Synthetase. Yes, synthetase. Because it required ATP. And then finally we have argininosuccinate. It kicks off the fumarate and we get arginine. And that one was, I think someone already said it. What enzyme did that one? Had a common name. It's the argininosuccinase. And that's how we get arginine. Okay. So, I think this is a good place to end for today. So what happens whenever arginine concentration is low in mammals? This is one time that we can, remember, primarily, most of the time in this chapter we're focusing on bacteria, but I would point out times whenever there's a difference, major difference between mammals and bacteria. This is one. So we can actually scavenge from Prolene to make arginine, if we need to make arginine. Okay, so let's say that we're not getting it through the urea cycle derivatives quick enough. So if you remember, back in Prolene, one of the intermediates was this P5C, this Prolene. It was non-enzymatic. Well, whenever it opens up, it's that glutamate semialdehyde. And so we can actually take another enzyme, and it takes this glutamate semialdehyde and alpha-ketoglutarate and glutamate, and transform the glutamate semialdehyde into ornithine. Okay, remember, and then ornithine is what can go on into the urea cycle to ultimately make arginine. So what enzyme, or what class of enzymes can do this? So we've seen this kind of thing before. Transaminase, okay, so an amino transferase. And so what specific amino transferase could this be? I can see where you could have a couple of alternatives for naming it. If you want to name this amino transferase, what's one possible name? Pardon? It's ornithine ornithine amino transferase, yes. Okay. And this, they call it the delta. That's the position where the nitrogen got added. It's on the terminal end, not the end that we are used to seeing it with the other amino transferases. Get ready to sneeze. Sorry. Allergies are horrible today. Okay. I've already had to run to the doctor once. Okay, not for me. I woke up and one of his eyes was swollen shut. And so, yeah, he got conjunctivitis like overnight. So. Okay, so. Pardon? The delta? Okay. That's positioned just like alpha, beta, gamma, delta. So that lets you know, remember this is the C alpha, beta, gamma, delta. Lets you know the position that the amino group is If you just called it ornithine amino transferase, I'd give you credit. That's the important part. Okay. So let's go to the next group. That finishes up that entire group of amino acids that we've talked about so far. So the next group are those that are based off of 3-phosphoglycerate. Where have we seen 3-phosphoglycerate before? Let's go back. It's glycolysis. These are the glycolytic ones, the glycolytic amino acids. So we're going to go from 3-phosphoglycerate to serine, and from serine you can make glycine and cysteine. Okay, so yes, that is glycolysis. Oh. I thought they were getting a little excited about hearing the Beatles earlier. All right, so let's look at this. Okay. Give you a moment to look through the process. This is the entire process. I would expect you to be able to work your way through all of this. It's not anything new in that sense. These specific enzymes are even new, but we've seen these classes of enzymes before. So if we start with 3-phosphoglycerate, I would expect you to know how to draw 3-phosphoglycerate because you draw glycerol, which glycerol looks like what? It has how many carbons? 3. And what makes it glycerol? There's an alcohol in each carbon, and then the 8 just means that you transformed one of them into carboxylic acid. And so it can only be one of the, it's carboxylic acid, this is one of the ends, right? It doesn't matter which end, because it's identical. We usually draw it at the top. And then the 3-phospho means the other end gets phosphate on it. So that's why it's not difficult. Because by default, if you think back to organic chemistry, the carboxylic acid becomes carbon number 1. Okay. So in the very first step, we have 3-phosphoglycerate becoming 3-phosphohydroxypyruvate. It's a mouthful. It utilizes NAD. You do get an NADH out of it. So what class or classes of enzymes have we seen that can do this? What? It's a dehydrogenase. And so what, if you're going to name it, what could you name this dehydrogenase? Bingo. So, if you didn't put, if you put 3, that's perfectly fine. Yes. Okay. Phosphoglycerate dehydrogenase, it doesn't matter to me if you put 3 or not. All right, but yes, it's phosphoglycerate dehydrogenase. So now we've made the 3-phosphoglycerate. Now we made the 3-phosphohydroxypyruvate. Woo, that's a mouthful. Okay. All right. And so if we look at the second step, we take glutamate, it transforms into alpha-ketoglutarate. So the only difference between these two now is that we are transforming it into an amino acid. Okay, so that was the keto version, and that's the alpha amino acid. So what class or classes of enzymes can do this? It's an amino transferase. So what amino transferase is this one? It's got 2 possible names. If you just use it based off of the logic that we've seen so far, typically it's named after the amino acid end. So knowing that, what would we call this one? It is, you can reverse it. I mean, it is an amino transferase. It's just not showing it being reversible here. So what could we name this amino transferase? Pardon? No, it's an amino transferase. So if you compare the 2 possible products, remember it is reversible. Amino transferases are reversible. We have the 3-phosphohydroxypyruvate and we have the 3-phosphoserine. Which one is the amino acid? 3-phosphoserine. So we would call this one. Yes, it's the phosphoserine amino transferase. Now if you put the 3 there, that's fine. It's not. That is what phosphoserine looks like. Okay. So now we just need to dephosphorylate to transform it into serine. Okay, so we have 3-phosphoserine, and we're removing this phosphate. It's in the form of inorganic phosphate. So water comes in and smacks it, pops it off, okay? So it's been a little while since we've talked about the various phosphate containing or phosphate utilizing enzymes. So what class or classes of enzymes can do this? Think back. If you need to, it would be a good review. It's a good review. We could just review all those enzymes. There's at least 3 that I can think of off the top of my head. Classes of enzymes that utilize phosphates. What are they? We have phosphorylases, and what do they do? They add phosphate groups without using ATP, P2, blah, blah, blah. So those are the phosphorylases. What are the other 2? Kinases, which they transfer the phosphates utilizing some of the NTPs. What was the third group? Phosphatases. And what do phosphatases typically do? They remove phosphates in the form of inorganic phosphate. Technically, if it's an inorganic pyrophosphate, they call them pyrophosphate. But okay. But yeah, so knowing that now, what class of enzymes can do this right here? It's a phosphatase. And what phosphatase is it? Don't overthink it. What are we phosphatasing? Phosphoserine. So put it together and you get, bring on home now. Thank you. Yes, it's phosphoserine, phosphatase. Sometimes I have to be cheerleader. No wonder my iron levels and energy levels are low. It's wear me out. This next one is really the hardest. So we made serine. It's just one step forward for us to make glycine. They're happy. This uses that H4 folate. What does anyone know? That's the way to abbreviate. What's that abbreviation mean? What is H4 folate? 4. What's a good prefix for 4 in science? What? Tetra. So this is tetrahydrofolate. It's a compound we needed to have, a cofactor. So this is tetrahydrofolate. You get a methylene folate out of it. It utilizes PLP and a water. Okay. Alrighty. So I'll just tell you right now. This one is named for the reverse reaction for going from glycine to serine. What? If you're going backwards, and I don't know historically, maybe it's because they found glycine first, and they found out that in vitro I have no earthly idea what the story is. But if you're going from glycine to serine, what's the only difference between those two? It has an alcohol and what else? Carbon. And a carbon. If you put those together, what are those called? Where you put that CH2OH group together, what are those called? No, it'd be CH3OH, but you're really, really close. Whenever it's a functional group that's hanging off. So you guys remember, you don't probably take a lot about it in organic chemistry because usually, at least in organic chemistry 2 level, the alcohol gets priority. You know, the alcohol would be for the naming. Of course, here it's not. It would be the carboxylic acid. So, what is it called when the alcohol is not the priority? Whenever it's the thing that's hanging off, we don't call it. It doesn't get the ending. It's a prefix. Hydroxy. It's a hydroxy. So whenever it's a CH2OH group hanging off, so you remember what that's called? It's not called methanol, but it's called hydroxy. Okay, it'd be hydroxymethane, but when it's attached to something else, it's hydroxymethyl. And so this was a hydroxymethyl transferase. So it's serine hydroxymethyl transferase. So I wanted to walk through the logic. It is a transferase enzyme. One never goes into that. But it's the most difficult part of all of that. Are there any questions so far? I said making serine is easy, and it's just one step to glycine. You just have to remember it's the transferase enzyme because it's in for backwards. The next part, cysteine. the book actually divides it up because we mammals do it so differently than plants and bacteria. So I want you to know the big scheme, just the basics for plants and bacteria, but I want you to know the details for mammals since that's the one that's more medicinally, biomedically relevant. And so this is the big scheme for bacteria and plants for cysteine. Okay. So I want you to know a couple of things. One is it's actually fairly complicated. But two, what amino acid is used to make cysteine? What amino acid is required to make cysteine? Serine. Okay, so it's derived from serine. Because usually if I ask this question, if I ask it with respect to bacteria and plants and mammals, it's like a compare and contrast. I'm not going to ask you each step of the enzymes and blah, 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 blah. Okay? So it starts from serine. And you should know what cysteine looks like. But the real difference has to do where the sulfur comes from. Where we get our sulfur for cysteine is very, very, very different than where they get their. Where do they get their sulfurs from? You know, it's highlighted. We're supposed to, it's highlighted. Their sulfur entered into their, what's that, metabolome or whatever, their environment, in what form originally? So all the way from the beginning, it came in the form from, yes, from a sulfate. Sulfate turns into sulfide through a series of reductions. But yeah, so there's the sulfate, and then it gets incorporated into cysteine. Okay, so that's what I mean by knowing the basics. Serine is required to make cysteine. The sulfur came from sulfate, maybe at a sulfide, but yes. So that's what bacteria and plants do. But I want you to know for humans, because like I said, this is the one that's very relevant to us. What amino acid or amino acids are important for making cysteine for us?
Is it complex? The way you said amino acids.
Well, we're going to get there. Oh, just start at the beginning. Don't overthink it. What amino acid is required for us to make cysteine? Serine. That's one that's on the right. The right. What other? There's another amino acid that we use. Methionine. It's not listed here, but that's what makes homocysteine. Okay. So if you look, our sulfur comes from methionine. It doesn't come from sulfate. It's going to come from methionine via homocysteine. We'll talk about where the homocysteine comes from with methionine later on. I haven't checked the most recent edition, but the older editions, I would do it in a little different way. The homocysteine is a derivative of methionine. If you look at what's the only difference between homocysteine and methionine? Yes, the methyl group is missing. So whenever methionine got a methyl group because it's a sulfide ending, whereas homocysteine is just like methionine, it's missing that one carbon on the end. So that's why it's called homocysteine. It's just like cysteine, it has an extra carbon. All right. Okay. So that's why we actually have to have 2 amino acids in order to make cysteine. But there are much fewer steps. Right. So notice we have PLP. We've seen PLP over and over and over again. Pyridoxal phosphate, if you forgot what PLP stood for. All right. So what class or classes of enzymes can take that homocysteine and that serine and smush them together? A what? It's a synthase. Okay. And what are we synthesizing? It's cystathionine. So in the beta, just let you know at what position, but yes. You just call it cystathionine synthase. I give it to you. Beta just lets you know the orientation. What carbon can you take? Because technically there's more than one end that can be. Okay. And then the last step, once again, it uses PLP. It takes a water and it deaminates it, but it is not a deaminase. Okay, so that's why I wanted to. And so we get 2 things out of it. We get alpha-ketobutyrate. Does anyone know what is alpha-ketobutyrate? What kind of compound is it? Like if this was organic chemistry, not forget about biochemistry. That was organic chemistry. What kind of compound is alpha ketobutyrate? It's a carboxylic acid, and it's a what other functional group does it have? Ketone. This is one of our ketone bodies. Like if you go to ketosis, or if you're on a ketone diet, you know that kind of thing. This is a ketone body. Okay, so it's named after the fact it's got ketone. It's also an alpha keto acid, just like what we've seen before. It's just that this one is not an alpha keto acid for an amino acid that's used to make proteins. I guess in theory you could turn this into amino acid, but we don't. It's not one that makes a protein. Like it's not valine or leucine or something like that. Okay. But then we get cysteine. So this is a ketone body, and we get cysteine. Okay. So what class or classes of enzymes can take something big and break it apart? We don't use this class very often. Because it's not just deamination. If usually deamination just means that you're only removing the amine group. Here we're not only removing it, but we're also breaking it into two smaller things. What do you remember back in the classes of enzymes that we had? What one it is that breaks things apart? What? Technically, hydrolase would be a subcategory. It's a bigger group. It's like one of the big classes.
Does it have cobalt? Catapult in the name?
Do you remember? I mean, like I said, I think we've only seen this name like once or twice all year long. Forget about this semester, all year. Not ligase, but you're really close. Lyase. This is a lyase. Literally, that means you're making or breaking the groups. Usually it means through the formation of carbon. There's carbon sulfur bonds, carbon nitrogen, carbon oxygen, and I think carbon and phosphorus technically. I don't remember the little nuances there, but and the gamma once again just lets you know what position. It's cystathione gamma lyase. Remember, in biochemistry and biology in general, we use the Greek lettering system for locants a lot more frequently than they do in organic chemistry, with the exception that organic chemistry likes to use them for alpha beta, like when they talk about like unsaturated, saturated bond, they don't use gamma and deltas and stuff that often. I don't know why. Okay. Oh, so and then there. We've already answered this, but I didn't answer those questions as we went along. Sorry. Okay, so are there any questions on how to make cysteine? Remember, the big difference, if you want to do a comparison and contrast, one way that the two major systems are similar is that they both utilize cysteine. I mean, they both utilize serine. The big difference is mammals also require get our sulfur originally from methionine via homocysteine. Plants and bacteria gets their sulfurs from sulfate.
So that's what we need to know.
Yeah, like the big C thing. I mean, you need to know these two enzymes, but I wouldn't ask you like the various reductases on a plant. I mean, in the sense of memorizing it. Like if I gave you lit it all up and had to use the logic on trying to name something, but I don't usually do that. All right, so the homocysteine, we've already talked about this. It comes from methionine. So we have SAM, or Adomet. Remember the one that is S-adenosylmethionine. It's a very important cofactor. If I give you a picture of what adenosine looks like, you could draw this, because the S is just the locant, means the adenosine. The YL just means that it's hanging off. So the adenosine is hanging off the sulfur of methionine. You put it all together, you can draw it. It is used in multiple pathways. But it's also important. Oh, and homocysteine, by the way, homocysteine. It's used for, and homocysteine, by the way, also has a 3-letter code. It's HCY, just in case I draw it that way. And there are various disorders. Homocysteine is also a marker for metabolic disorders as well. So there's homocystinemia, homocystinuria. Homocystinemia means what? Where's the homocysteine? The higher levels they're in the blood. Homocystinuria would be in your urine. Yes. I guess technically you have hypo and hyper usually when we're talking about for metabolic biomedical disorders, we're talking about hyperhomocystinemia or hyperhomocystinuria. It is a marker for like heart disease and various defects. Okay, so this is from chapter 18. It's a good review. Like I said, you should know this. It should make sense. We have methionine. It takes ATP. The big difference to make SAM or Adomet is that the sulfur, when it attacks this carbon. Usually when we see ATP, we're doing a phosphorylation or a pyrophosphorylation reaction. But this one, we're actually directly attacking the A, meaning the adenylyl group. Okay. And if you notice, it gives off three phosphates, but when they hydrolyze down to inorganic pyrophosphate and inorganic phosphate. So this is what Adomet looks like. Methyl transferases are the enzymes that do this. But that's why there are lots. Okay, it's just a class, technically a subclass of groups, and they transfer the methyl groups to wherever what needs to be methylated, and we get S-adenosyl homocysteine. And then there's a hydrolase that comes in and kicks off the adenosine group. And so that's how we get homocysteine. This homocysteine is what can be used if we need to make cysteine. It's used to make cysteine. Okay, if it's not needed for that, then we actually use methionine synthase to remake the methionine. Like I said, this is technically a review. Okay. Are there any questions on this? So SAM, when I say know the normal function of SAM, it's used for methyl transferases. It's the way that our body has to add methyl groups to lots of different things. Okay. We're only going to spend like a matter of a few seconds. The next slide, let you know, matter of a few seconds on it. It's literally review. We reviewed it the last class. We have reviewed it at least 20 times throughout the semester, I bet. Because we've seen it over and over and over and over again. It's alanine and aspartate. What enzymes or classes of enzymes are involved to make alanine and aspartate? Those, what kind of transferases? Amino transferases. So alanine comes from what? Pyruvate, and aspartate comes from oxaloacetate. So that's what I mean by those. I just want to keep referring to. Okay, so now we're going to talk about asparagine. That's why I just wanted to briefly. I want you to think about this for a moment. I didn't want to give away too many answers. That's why let's give you steps. We have asparagine. There are 19 other amino acids. What amino acid or amino acids is asparagine very similar to? Aspartate, and what's the other one? Glutamine, and glutamine is very similar to glutamate. Okay, so knowing those four, this is sort of like the SAT kind of questions, Glutamate is to glutamine, while the aspartate is to asparagine, that kind of thing. Okay, so knowing what you know about those, you should be able to put together the idea of where asparagine comes from. Okay, so if we work backwards, we have asparagine. Like if we start with glutamine, remember glutamine's from what chapter? The chapter starts with blending together, probably 18. Glutamine came from glutamate, all right, and glutamate comes from, what's its keto acid? Alpha-ketoglutarate. All right, so using that logic, asparagine will come from ultimately oxaloacetate, right? Because it comes from aspartate, aspartate comes from oxaloacetate. So that's why I said, see, you already know a lot of this. If you just sit there and don't overthink it. I should say don't. I've seen people like second guess themselves. Okay. So I've numbered these. Give you a moment to look at it. So that's why I wanted to. I think it through. Okay. So let me actually go backwards to make sure that I did these in the correct order, because I have been known to number them and then do the slides out of order. All right. So if we look at number 1, what? So we're starting with oxaloacetate to aspartate. What compound is number 1? I know it's not the pretty, it's pixelated, and it's Google Images. So it's glutamate. Okay. What compound is number 2? Is this box number 2 right here? And someone said it. It's alpha-ketoglutarate. So then putting all this together, going from oxaloacetate to aspartate, what enzyme is that? It's what? It's an amino transferase. And then if you're going to be the specific amino transferase, what specific amino transferase would it be? It's aspartate amino transferase. Like the vast majority of the amino transferases, they're never named after this. Remember, this is utilized for all amino transferases. It's usually named after the amino acid. That's the other amino acid. So yeah, this is aspartate amino transferase. So we made aspartate. So now we have compound number 4. Okay. What is compound number 4? You have seen it before. It may look a little different. It's pixelated. It's glutamine. Okay. What is compound number 5? It's glutamate. So now the only thing that we're doing here is we have aspartate, and we are adding the terminal nitrogen. We do have to say which end, because technically, you know, it's got 2 carboxylic acid ends. And so we're adding it to the alpha, beta, gamma, the alpha, beta, gamma, delta, the delta, gamma position. Right? It utilizes ATP. So remember, we've seen this kind of thing with glutamate becoming glutamine as well. Remember, it required ATP and everything else. What classes of enzymes can this be? When this was a glutamate becoming glutamine, what was the name of that enzyme? It uses ATP. It was glutamine synthetase. And so what is this one? There you go. What's an arginine? It's asparagine. But yes, I knew what you meant. See, hopefully the logic to it you've been getting throughout the semester will help you through a lot of this. There are diseases associated with a lot of these metabolic pathways with respect to amino acids. Okay. All right. So, building off the asparagine can actually be degraded, just like we had glutamine can become glutamate because it can be degraded. So given what you know about glutamine becoming glutamate, you should be able to name asparagine becoming aspartate. And then I'm going to show you about the catalytic triads in just a moment. What's the name of the enzyme that goes from glutamine to glutamate whenever it degrades? What? It's glutaminase. Okay. So what enzyme, once again using that logic, what's the name of the enzyme that goes from asparagine to aspartate? It's asparaginase. Mm-hmm. It's the one that's going to remove that terminal nitrogen in order to make it aspartate. This one's going to, whoops. Asparagine. I say asparaginase. I guess we can call it asparaginase. It's a snacks and thing. Yeah. But it's named after asparagine. Okay. Mm-hmm. Say it with confidence and people go with it. Say it with a British accent, they think you're really smart. say it with a Canadian accent, they think you're not. I don't know. Or, you know, from Mississippi or something like that. All right, so this one, we've talked about catalytic triads before. The one that we went into the greatest amount of detail last semester was the one that had the aspartate, the histidine, and the serine. You remember the aspartate would take the either take or give the nitrogen from the histidine would take or give the nitrogen from the serine to make it a good nucleophile. This one's got a little different. It has the catalytic triad here. That's one of the reasons why they like to talk about this. So what we have here is we've got a threonine that's important, tyrosine, and a glutamate. This threonine right here is just holding it in the correct location, right? So the step one is you're going to form a tetrahedral intermediate using that first catalytic triad. So this will take the hydrogen from the tyrosine takes the hydrogen from the threonine becomes a great nucleophile to smack the carbon of the amide, okay, which will break to make the tetrahedral intermediate, and then you get collapsed the tetrahedral intermediate, and the nitrogen will leave. See?
So does the tilde mean that there's probably more on the rest of the
Well, that little
line?
squiggly line, yes, it just lets you know that they're only showing you the R group, the part, the business end, and that there's a lot more. This is in the catalytic, the catalytic side of the enzyme. Of course, now it's covalently attached, so you got to remove it. And so the way that we do that is we're going to activate a water molecule in order to remove that tetrahedral intermediate. So it's a different tetrahedral intermediate that comes along. So we have the first tetrahedral intermediate, and this one we have a lysine, aspartate, and threonine. I just want to show you this, and it's actually a water molecule. That's the nucleophile. But we have a lysine, and this aspartate. This helps take away the nitrogen. So this one will grab the nitrogen from the threonine. Threonine grabs the nitrogen from the water, so that we can acting more like a hydroxyl anion, which can come and smack the covalently attached. You form a tetrahedral intermediate, and you get collapse of the tetrahedral intermediate, and by doing so, it releases the previous threonine from the first tetrahedral intermediate. Okay, and so that's why, and I wouldn't ask you to draw all this one out, because I'm trying to draw this all out in a little spot in the. But I mean, I can give you examples and ask you, you know, from general base from last semester, you know, what would happen if we do this and that kind of mutation, that kind of stuff. I probably wouldn't ask you to draw all the electron pushing out just because of the fact that it would take more than a page. So and then we get the final scenario where you get release. So now we have both of the catalytic triads are released. We have the glutamate product. I'm sorry, the aspartate product, which can then go on and do whatever we need for the aspartate to do from the various things. Okay, so there are medical implications for this. I just picked one, like I said. You can have entire classes, not just like individual lectures, but entire courses on these metabolic disorders and amino acid disorders and stuff like that. I usually just try to pick some. For whatever reason, ALL, which is acute lymphoblastic leukemia, they require asparagine for their growth. That specific type of tumor requires asparagine. And so if you know this, if you know that you've got this tumor in a child, it's going to require asparagine for growth, what kind of treatment can you possibly give in order to help this child out? We want to get rid of asparagine. What's one of the possible treatments? How can we get rid of asparagine? Just knowing what we've talked about so far.
You increase the asparaginase.
Exactly. Good job.